Textor, Charité Universitätsmedizin Berlin, Max Delbruck Center for Molecular Medicine. Refining the targets for T-cell receptor gene therapy.
Hello everyone, so I'm also going to talk about T-cell receptor gene therapy, but I'm going to focus more on the mechanisms that underlie tumor rejection versus tumor escape. So tumor neo-epitopes are presented on the surface of the cells in the context of MHC class I, and these peptide MHC class I complexes are then recognized by T cells through their TCRs, and these tumor-specific T-cell receptors can then be used for T-cell gene therapy by expressing them on the patient-derived T cells and then infusing those T cells back to the patients. There are two major factors that should be taken under consideration when designing optimal T-cell receptor gene therapy. First, the target epitope has to have high peptide affinity for the MHC class I. And second, the T-cell receptor has also to have a high affinity for the peptide MHC. However, despite these precautions, tumors still often recur after therapy. So we have investigated this further, and we have seen that the neo-epitopes require additional evaluation. So in brief, the tumor antigen which contains the neo-epitopes needs to be degraded by proteasomes to smaller peptides, and these peptides are then transferred to the endoplasmic reticulum, and if they are of the correct size, they can be loaded onto the MHC class I and presented to the T cells. But often they are just elongated precursors which need additional trimming, and this is done by an enzyme called ARAP1, and once they are of the correct size, they are loaded onto the MHC class I and presented to the T cells. In our recent publication, we have shown that if we target epitopes which are already of the correct size, we get tumor rejection, but if we target those that are elongated and need trimming, then we get tumor recurrence. And these tumors escape because they downregulate the function of ARAP. So we would like to introduce a third factor which is necessary for designing optimal T-cell therapy, and this is to target targets which are ARAP independent. And how we're going to implement this into the clinic, we are going to take tumor biopsies, sequence for the neo-epitopes, and then test those neo-epitopes whether they have high peptide MHC affinity and also that they are ARAP1 independent. After that, we're going to immunize the mice that were introduced by Matthias, and from then we are isolating the high affinity TCRs, and we engineer patient-derived T cells and infuse them back into the patients. Thank you for your attention.